Greeting to Dr. Hasmaliana. Today we are going to present our project Omic Technology. We are from Group 5. We are Nur Amira, Nurul Izzati, Shakira Sufaira, and me, Wong Rui Fang. What is Omic Technology? Basically, Omic Technology is the part of biotechnology which analyzes the structure and function of the whole makeup of a given biological function at different levels. Omics refer to the collective technology used to explore the rules, relationship, and action of the various types of molecules that make up the cell of an organism. These technologies are primarily aimed at the universal detection of genes, which genomic, mRNA, which refers to transcriptomic, protein, which is proteomic, metabolite, which refers to metabolomic, carbohydrate, which refers to glycomic, and lipid, which refers to lipomic, in a specific biological sample to study the mechanism, interaction, and function of cell population, tissue, organs, and the whole organism at the molecular level in a non-target and non-biased manner. Why omic? Omic technology are required at the several applications. This technology is important for human health. For example, genomic is used for diagnosing a disease, forensic testing, and newborn screening because genomic is used for gene therapy and gene testing. Another example is proteomic for heart disease or HIV. Toxicity assessment, vaccine development, pharmacology in drug discovery is also an application for other omic technologies. Biomarker is one of the applications for proteomic and also glycomic.
What is glycomic? Glycomic is the study of sugar or carbohydrate chain in living cells. Carbohydrate chain, glycan, form on cell membrane of every living cell. Carbohydrates can also attach to protein and lipids to form more complex molecules and key to almost every biological process. For example, when you injure yourself, glycans help to send a signal telling white blood cells to gather the injury site to fight the infection. And for the blood transfusion, the type of glycan on the surface of blood cell will determine the type of your blood. Why glycomic technology is required? Glycomic technology is important as glycans are involved in every human disease. For example, the first one, inflammation, which underlies cellular damage associated with most diseases, including infectious disease and metabolic disease, is regulated by glycans. For example, the multi-step leukocyte adhesion cascade. The second one, immune system regulation. Glycan play an essential role in the regulation of the immune system, both innate and humoral. For instance, major blood types in humans are determined by the glycans. The third one, vaccination. Glycan play a key role in infectious disease and in vaccine development. For example, recognition of bacterial carbohydrate and lectins. And the fourth one, cancer progression and biomarkers. Glycans play a direct role in cancer progression and as a biomarkers for early detection of cancer. Next, number 5, diseases. Both intracellular and extracellular glycans, for instance, glycosylation, underlie molecular mechanisms leading to chronic diseases such as diabetes and Alzheimer's, as well as play important role in diabetic cardiomyopathy. Number 6, bioactivity of drugs. Glycan play a critical role in the bioactivity and the pharmacokinetics of drugs and development of new drugs. And then lastly, number 7, human development. Genetic diseases of glycosylation in inborn errors of glycan metabolism illustrate the critical roles of glycans in human development. In conclusion, glycomic technology needs to be explored so that we can benefit the pros and reduce the cons.
Glycans as biomarkers for diseases. Glycosylation alters cellular phenotypes and it is crucial in the genesis and progression of pathogenic diseases including cancer. Interpretation of the role of glycosylation in biological processes is difficult to ascertain due to their intricate multi-branch structure and multivalent nature of recognition. Biomarkers can be used to predict the probability of developing a disease, as well as to aid in the initial diagnosis and staging of illnesses and to track disease development and the effect of treatment. An excellent tumor biomarker would enable clinical choices to be aided by a simple blood test. For the tools involved in this is Glycobase, a database containing the HPLC elution position for over 352 alpha-beta label and glycan structures. Next is AutoGU, resources for interpreting HPLC glycan data, and the last one is EuroCARB-DB, a database containing glycan structures and their biological context. As for the equipment involved in this is high-resolution mass spectrometry, high-performance liquid chromatography, lectin microarrays, and capillary electrophoresis. 3 stages involved in the technique. Stage 1 is sample preparation. For the first step in the stage 1 is glycoprotein immobilization, and then glycan release with PNGASF, and extraction of glycans. Next is fluorescent labeling with 2-alpha-beta, and the last one is 2-alpha-beta glycan elution. Next stage is chromatography. The first step in chromatography is HPLC separation of undigested samples, HPLC separation of samples digested with exoglycosidases, and the last step in stage 2 is wax HPLC separation of undigested samples. As for the last stage, which is stage 3, analysis of results, the first step involved in this is integration and quantification of relative levels of glycans. Next is interpretation and identification of potential biomarkers. The third one is structural assignments based on GU values and comparison with glycobase. The last step in stage 3 is statistical analysis to confirm significance of glycosylation alterations.
Assalamualaikum warahmatullahi wabarakatuh, and a very good day. My name is Nurul Bali. I will continue the part of glycans as biomarkers for disease by glycomic technology. First of all, when the glycomic technology is useful for glycan as biomarkers. According to Adam Schiff, glycan biomarkers in the detection of cancer. For the alteration in protein glycosylation, a common feature of tumor cells and may affect any type of cell glycoconjugate such as glycans and glycans on glycoproteins, glycolipids, or glycosaminoglycans. So for why it's normally applied, the glycans are often found to be involved in almost all human disease in one way or each other, where they partake in morphological process either at multicellular level or at single cell level. For example, here is glycosylation chain as an alternative option to A153 for breast cancer detection. So basically, it involves especially in the cancer disease. So for who is involved in the application process is basically the breast cancer is mostly for females only. The serum and glycom analysis of whole serum from breast cancer patients has revealed the alternation of glycans.